So this video is about lab notebooks. Lab notebooks are really important. They're important in here, in this class, because they're a big part of your grade. But they're important in the real world, in the world of science, for a number of reasons. And they serve several purposes. One purpose for a lab notebook is the obvious one. It's a place to record what you did, what you observed, the results of your experiment. And you want to record that stuff often so that you or someone else can do it again, right? That's the whole basis of science, that it's reproducible. So a lab notebook is in some sense a recipe book that allows you to repeat experiments and store and share the information you need to repeat those experiments in the future. Well, there's another side to lab notebooks that is often less appreciated by students, and that's that it is actually a legal document. And so if you find yourself in industry somewhere, at a company, say, doing science, you'll find that they have very strict requirements for how those notes are taken. And you see some of this actually even in classes. I think our chem labs still do this, where you take notes and they have to be in ink, and you have to initial and date each other's notebooks. And all of that is actually connected to this idea that these notebooks are actually legal documents that have been used in a number of really important intellectual property cases to decide essentially really financially important things. Now, we're not doing anything in here that I anticipate having a ton of financial value, but it is still important to be able to keep a notebook at that level. What I found is it's really, really easy to keep a sloppy notebook. And let me just show you the next picture to tell you what I mean. I grew up, when I was a grad student, I took notes on brown paper towels. So I basically had them scattered all over my bench and my desk, and I would just scribble down what I was doing. And I did it in part because we were in a pretty casual lab. I could get away with it. My brain was still functioning back then. I had a pretty good memory. And I just didn't, we really weren't so uptight about recording things. And in hindsight, it was really kind of stupid. It doesn't take that much more work to keep a more proper notebook. And there were a number of times where I regretted my system. For example, if the brown paper towel got thrown out. A more traditional format is the center one, a paper notebook. It could either be a standard single-page paper notebook or one of those ones they use in chemistry with the, what do they call that stuff? Do they still have it? The stuff where you write on one page and it shows up on the next one? See, I told you my brain's messed up. Oh, it'll come to me. Anyways, technology has moved on, and so will we. So my favorite method for keeping notes is shown on the right. We will keep a digital notebook in here. The digital notebook will be kept obviously on your computers, and there's a real collection of advantages to keeping notes in that way. The biggest one from my angle is you can share it with me. You can say submit it to Canvas, for example, and I can read it anywhere. I don't have to lug around a box full of the standard notebooks. But more importantly, if I want to read it in 2 or 3 years, and believe it or not, that does happen. I refer back to student notebooks all the time. All the zebra mussel research I'm working on right now began as student projects many years ago, and I still look back at some of that data. So if we keep it digitally, I can keep a record of it and have access to it at all times. And importantly, you can too. Now, I don't know if you'll ever want to look back at your notebook for this class, but it's possible. I have actually had some students who've gone on into science careers tell me that they use their molecular biology notebook, for example, in their graduate research. So sometimes I actually teach you things that might be useful later on. It's still bugging me. What is that stuff called? Oh well. So yeah, let's not use the brown paper towel. Let's use the computer. So how are we going to keep our digital lab notebook? There will be one notebook submitted to Canvas from each group, so it is in some sense a group notebook, but I'll clarify that more in a moment. That doesn't mean that one person, you're all responsible. Let me say that. So it's a collective notebook. Now you're going to be given a lab each week, or a couple labs each week that are that has what I want you to do written up in a Word document. And you should know this. I hate busy work. So you should treat that lab as a scaffold that you build upon. In other words, you don't need to paraphrase. I'll give you a purpose for the lab. You can use that purpose. Now feel free to paraphrase it, but you won't get any more points. You're just going to add to the lab I give you, and you'll modify it. And I want to go through in very specific ways what I mean by that in the next few slides. But in the end, when you're all done, you'll have this Word document, a very large Word document, which is a collection of all the labs that you've conducted this semester. And as you know from the syllabi, you're going to turn these in several times during the semester, and what you'll do is just submit that as a single Word document, and that Word document will just keep growing. Now, as I mentioned a moment ago, everyone in the group needs to contribute to this notebook. I've had a few isolated incidents over almost 20 years now where I hear from groups that someone's not participating. So I've come up with a few ways to help deal with that. The first way is just don't do that. You're here to learn, so let's all try to work together. But what I do want to do to ensure that everyone gets an opportunity to work on their note-taking skills is we're going to rotate who actually records the observations each week and who drafts the, I'll call it the pilot notebook, the original, which then you'll all edit. I'll call that person the scribe. That scribe is the student who that week is taking the notes. And you will note in your notebook who the scribe is each week. So you put the initials down, who's the scribe. And again, rotate that. Again, two reasons. One is to give you all equal chance at taking notes, and the other is to deal with any issues if they come up about unequal contributions. The other way I want you to all contribute to this collective notebook is to share it in Google Docs. I can tell you I've been doing a lot of note-taking this summer. It's been good. I've been doing a lot of science and working with a number of people. And so I'm actually living this exact policy. I write up labs each week that myself and my technicians work on. I share it with them. I may start the experiment off, or they will, and then at various times throughout the week or weeks, we all make observations and contribute to the notebook. And we do that through Google Docs, and it's very effective. And I'll show you an example of that in just a moment. It's effective because everyone can contribute, edit, add comments, etc. So this is actually a screenshot from an experiment I performed a few months ago, or we performed. This is the data from that experiment. And what you see is the data is tabulated. This is a table. It shows you jars, and these jars are just labeled things like by letters, A, B, C, D, etc., and then some numbers because they're paired jars. Now those jars contain zebra mussels, and the zebra mussels are treated with various types of double-stranded RNA producing bacteria. So we're actually testing some of these RNAi constructs in this experiment. And so there's a code, there's a table somewhere else in this lab that tells you what A, B, C, and D, etc. are. Just for tracking it, we just decided it was easier in the lab to write A instead of the long gene name. Now you see what's going on here, that these numbers represent are the number of mussels that have attached to the jar. Because what we do is we put the mussels in, when we take them off of a substrate, like a rock for example, drop them into this jar after they've been treated with these bacteria. And what we're asking in this experiment is, do they reattach? We call it a reattachment assay. Do they stick to the jar side? So at time 0, none of them have. That's not too surprising. But then we monitor at subsequent times. In this case, we looked at 1 day and at 2 days. And so all those numbers represent the number of animals that were attached to the jar. Now, if you could see other pages in this notes, you would know how many total mussels were in that jar, and I believe in this case it was 10. So over on the right, you see my technicians' names. These are two great scientists. Lindsay Gengelbach and Margaret Donovan have been working with me all summer. And they have some really great lab skills, I can say that. So they must have had a good lab courses at some point in their undergrad career. And so again, a lot of what they've been doing is making observations. And so they actually put the numbers in for everything but 0 hour. You can see how it says SB for 0 hour. That was me. But then 24 hours, it says LGMD. And that's actually bad. I don't know who made those observations. We should have fixed that. What I was doing is saying, one of you two do it. I can't. And I could probably figure it out here. Yeah, I can. The comment tells me that Lindsay started, so LG. So she made those 24-hour observations. The 48-hour observations were actually made by Margaret. And you can see the areas that are glowing. If this were actually the Google document, you would put your cursor on there and then it would light up the various comments. And there's nothing that significant about these comments. I just wanted to make a point that Google Docs is a way for multiple people to have input into a single document. And of course, you probably know that already. Okay, so feel free to use Google Docs. I'd encourage you to use it both during the lab to track data like this, but more importantly, I think for you would be to use it after the lab when you're preparing the lab for submission. Right? You may have different ideas about how you want to present the data or the conclusions, and you could communicate that to one another through these Google Doc comments or through email or other tools. In the end, though, I get one document, and I would like it to just be the Word document. But do use these Google Doc tools as a way to track your contributions. And again, that comes up mostly only if there are issues with people saying that they're not, they have group members not doing their share. Okay, so next I want to go through the various parts of the notebook, the components, if you will. Every notebook will have a single cover page that will give some basic information to me. The information I want is the group color that I assign to you and your names. Right? Because I'm getting a single notebook, it'll be submitted to your group color, but I need to know who you are to give you your proper grades and to provide feedback. The second page of your notebook, and it might even be a third page depending on the course, is the table of contents. This is a listing of the various titles of the labs that we're doing and the page numbers that those labs are on in the notebook. Again, this is a reference so that one could go to the start of this notebook, just like in a paper notebook, and find where in the notebook the actual relevant lab is. Okay, item 3 is really the main thing I want to discuss now, and it is what are the components of each individual lab. Well, every lab will begin on a certain day. It'll have a date, and dates are really important in notebooks. I didn't note that in the previous example I gave you from our reattachment trial, but there were dates on there. And there are actually multiple dates, right, because we made those observations on multiple different times. Multiple different days. Carbon paper. That's what I've been trying to remember. They have notebooks with carbon paper. Okay. So after the date, there will be, or on the same line perhaps as the date, there'll be a title. Again, I'll give you a title for the lab so you can just use that. There's always a purpose. The purpose, why are we doing this lab? One of the things I really like about the labs I teach is it's not just to teach you something. I learn something from these labs. Right? We're doing real science, so there's a purpose. And I'll try to articulate that purpose to you, explain it in this narrative that I write. I'd like you to read it so you understand, and we'll also discuss it in class, because I really think it's important that you understand what we're trying to do. I mean, I suppose you could be a good scientific technician without knowing why you're doing the experiments, but I think that would be sort of vacuous. I certainly would want to know why I'm doing something. And I can tell you, working with the two great scientists I'm working with this past summer, they definitely wanted to know why. And then there were many examples or instances where them knowing what we were trying to do actually caused them to observe things that they probably wouldn't have if I just gave them instructions. So, it's best if you're all on the same page. After the purpose is materials. Materials are what are the things that you need to do this lab? What items do you need? Those could be test tubes or media or zebra mussels. And it should be quantitative, ideally. How many petri dishes do you need and how many zebra mussels? And the reason you have that grocery list is so that if you wanted to go back and do this lab over, or somebody else did, they could basically quickly go to the lab, look at the materials list and say, yeah, I have everything on hand to do it, or uh-oh, I can't start this lab yet. I need to pour some petri dishes or pour some, or go get some zebra mussels. So again, much like a recipe has a list of ingredients, this is the list of ingredients or materials you need to do the lab. I will provide that. Sometimes I miss things. It's not intentional, but sometimes I do. And so you, again, are responsible for not just using the list I give you, but keeping it up to date and adding to it if necessary. Next item on the list is procedure. This is often sometimes called methods. And this is what you're doing with those materials, right? What was the procedure of the lab? How did you start? What did you do next? So it's chronological. It's also quantitative, meaning, there's time intervals that you need to note, volumes, etc. This is the recipe. This is the written description of what you did, and it should be sufficiently detailed that I could hand it to someone, not say anything else, and they could do it exactly like you did it. So I will give you a procedure, and all I'll say is you are responsible. You can use that procedure, but you need to make clear what you actually do. So I have that point here in the bracket. Again, I'm going to give you a lab as a starting point with a procedure, but it might say something like, after mixing or treating the sample with DNase, incubate for 30 to 60 minutes at some temperature. Well, 30 to 60 minutes is not a single time. You need to tell me what amount of time you incubated. So if you chose 45, that should be what's in your notebook. So if I see something like 30 to 60 minutes in your notebook, I'll ding you a little bit because that's not exactly what you did, right? I may give you that, but then in the end you choose somewhere in there. The other point I want to make is that, we've got a few more sections still, results and conclusions, but before we get there, I just wanted to stress this. It's critical that every section in your lab is clearly labeled and in bold text. So it should say title, purpose, materials, procedure, results, and conclusions. I want you to do that because, first of all, it makes it easier for me to grade, right? I can make sure you have each of those sections. But also, it's really important that you put stuff in the right place. And the biggest area where there's confusion for this is typically these last two. There is a difference between a result and a conclusion, and it's really important in science that we make that distinction, because the result is what you observe, the conclusion is what you think it means. So let's talk a little bit more about results then. Again, these are observations you make during or after the procedure. So you do something and you wait, or maybe immediately you make some observations and you record them. So like the number of attached mussels in that previous example, those were results. They're typically collected after the procedure, sometimes during, but sometimes quite a bit after. As you saw with the reattachment screen, those were days after the procedure. And so again, you're going to need to add additional dates to your lab. It's not just when you start the lab, but things happen throughout afterwards. And this is kind of an important point, that your lab has many dates, and sometimes students get confused. They think that we start and finish a lab every week. I wish that were the case, but it's often not, or every lab period, I mean. It's often the case that it takes multiple lab periods to complete a single lab. Uh, the results should be tabulated or graphed when possible. Typically tabulated but graphing is possible. The trouble though is once you get into graphing, you can start changing the observations. So you remember the example I showed you a moment ago with the zebra mussel reattachment. Those were just numbers, the number of mussels that were observed attached. Now you could plot that. You could also take photographs, and I really like photographs. It's so easy now with everybody's cell phones, and they capture so much, right? A picture's worth a thousand words, and they're digital, so you can plop them right in your notebook. And the last thing I would say about results is that this is rather unusual in that we're often going to be doing the same experiment, in multiple groups in here. And it might be the same or there might be subtle differences, but regardless, I would like you to include all of the class data when it's possible, or at least a summary of what the other groups observed. Now, to do that, we're going to have to find ways to share our data. It's easy with digital stuff, or during class I might just draw a table up on the board, or you can come and type it into my computer. But bottom line is we'll find ways to share all the data with you. And having all that data is really helpful when you get around to writing your conclusion sections. I'll try to explain that with an example in a moment, but basically sometimes if all you have is your own data, you're left with a number of possible explanations. But when you look at the class data as a whole, you realize that some of those are more or less likely. So conclusions. Oh, I have the wrong thing highlighted there. I'll fix that. So conclusions are first of all, let me say this. The conclusion and discussion section, that's the section where students typically need to work the most on. And to say it another way, I guess, that's where you tend to lose the most points. And that's because, again, this isn't provided. You're given a lab that has a title and a purpose and methods and materials and stuff, but you don't have conclusions. Those are yours to write, and you should do it as a group. And you're typically going to be working on those outside of class. So in other words, in class, you'll be making the observations. Outside of class, you'll be manipulating those observations and then drawing your conclusions. As I said a moment ago, you're not always going to have conclusions every lab period or every week. Sometimes some experiments take multiple lab periods or multiple weeks to give you before you can make a conclusion. So don't panic. Don't feel like every date in your notebook or every time we get together you need to have a conclusion. Third bullet point on the right there, it says includes calculations based on data. So data is what you observe. If you start taking the numbers that you observe and doing things to them mathematically, that is no longer data. That is now a calculation based on the data, and so that should show up technically in your conclusion section. I think what I'll do is insert a second slide after this to make that distinction. Okay, so the next point, bullet point 4, discuss your group's data in relation to the rest of the class. So again, you're going to have all this data, not just your own groups, and that's really helpful in terms of the most important part, I would say, of a conclusion section, which is troubleshooting. I absolutely guarantee you that this semester we are going to have to do some troubleshooting. What do I mean by that? Basically, I mean that our experiments are not going to work the way we expect them to the first time. That shouldn't trouble you, by the way. That's normal in science. I've been working with a number of scientists for many, many years, and I have never met anyone, and I've worked with some really excellent scientists. I've never met anyone whose stuff works the first time every time. It just doesn't happen. Right? What separates great scientists from poor scientists is not that they do perfect experiments the first time. It's that they are good at figuring out what to do next when an experiment doesn't give the expected result. And typically what you want to do is become a detective. Right? So in your conclusion section, you're going to write out all the possible explanations for any unexpected outcome. Right? So just make a grocery list, brainstorm, think of all the possible things you could think of that could explain your result. And then once you have that list, you want to go through it one at a time and say, okay, I think this is a likely explanation, or I think this is unlikely, or I can even rule it out. And again, I know I'm going to have an upcoming example to try to explain this, so I won't go into that further because in a moment I'll try to explain that more. The last thing you should do in your troubleshooting section is describe basically things you wish you had done. What changes would you make to the lab or controls would you add so that in the future you could be more conclusive? Right? If you weren't able to draw the conclusions you needed to from this lab, what would you do differently next time? And again, that's what really separates great scientists from less great ones. They figure out what to do. They respond to poor outcomes with changes that make the next round more likely to succeed. And the last point I have is that this is the point where you should use statistical analyses if that's possible, right? If we have replicates, for example, you can do things like averages, standard deviations, and chi-square tests, whatever. Okay, so I stuck this slide back in when I realized I was having a little trouble explaining the difference between data and conclusions. On the left, you just see that data table I showed you earlier with the various muscles that were treated in different ways and how many muscles attached to the jar. What I did is from that data, I then calculated the percentage reattachment for each of these jars, remembering that these jars are muscles that have been treated in different ways. And it turns out A had no treatment, so that's the left pair of bars here. The B's jars were treated with a control strain, which makes a double-stranded RNA to GFP. There's no green fluorescent protein in zebra mussel, so that's just a random double-stranded RNA. And then these C, D, E, etc. are other, those are gene targets from zebra mussels. And there's two bars for each treatment because there's two data points, right? We have 24 hours and 48 hours. So what you see here are the percent reattachment at 1 day and 2 day for the various treatments, and then the error bars represent the variation, the standard deviation between the two observations, between the two jars. So your conclusion might include a graph like this, but then you're going to have a bunch of text, right? Because the underlying question here would be, what does this mean? Did any of these treatments disturb reattachment? Do we have a statistically significant change in reattachment between when we just treat any of these zebra mussels with these various double-stranded RNAs? And again, that would be compared to these two controls. And I think you could see with these error bars, unfortunately, the answer was no. The most hopeful one was RPN8 here, and it's hopeful because it stayed low. That's one reason. Most of these other ones are going up over time. It had very little variation, but because we had so much variation in the control, we couldn't say with confidence that this is a real difference. So that would be a conclusion. We can't say with confidence that there's a real difference, but now what would you do differently? How could we reduce the variation in this sample? And so one possibility would be to use more mussels, more jars, try to get the N value higher with the hope that that would lead to less variation. There's other possibilities as well. We could perhaps treat with more double-stranded RNA containing bacteria or more treatments. There's actually a lot of different things we've been considering for ways to make this effect more pronounced, if it's there at all. And so I won't belabor this particular one, but instead I'll go through a more simple a simple example of a lab notebook in the next few slides. So this is a sample lab notebook, a lab of a type that we may very well do in here early on. This happens to come from my molecular biology class a few years ago. So there's the cover page. This happened to be the orange group. I anonymized their identities. And so page 2 shown on the right here is the table of contents. There's a listing of various labs. Actually, we had like 50 labs or 30 labs or something, so this is just the first 9. And it was probably due at this point. And then you see the page numbers. So then the third page then would be lab 1. That's where it begins. And so here's their entry for lab 1. Just grabbed a screenshot of it. Oh, by the way, I did have permission from them to share this. Actually did quite a nice job with their lab notebook, but I didn't pick the best notebook I've ever found. I thought that might be less useful from a teaching standpoint. This is a pretty good one. And so let's start with what I like about it. I tried to highlight that in green. So here's their title, Lab 1, Transforming Competent E. coli. Got a nice date here and the scribe. Remember I talked about the scribe, so definitely put that stuff in your lab notebook. Purpose, procedure, results. I can't tell if those are in bold or not. I'd like them to be bold, so good start, right? And there was a conclusion. I'll show you that on the next page. What's missing? Yeah, if I was more clever, I'd have some sort of quiz or something here. But bottom line is there is a section that's missing, right? It's materials. So they didn't have a materials list. So that would have been one spot I could ding them. And so what I've got on the right here are things, basically places where they could have improved upon this lab. And you should know this. I'll do this for you guys too. I will give you comments on your notebook. I won't just give you a number. And the first time I'll try to be generous. I might actually just say, oh, you missed the materials section. Next time, keep it up and I'm going to really hit you hard. But they definitely should have had a materials section. This is just a partial list of the things you need. You need a plasmid to do a transformation. You need competent cells. You need some broth, some selective plates. Again, this may be all Greek to you, but when we do a transformation, I'll try to make it more clear, because what you're doing here is you're putting a piece of DNA called a plasmid into a microbe, in this case E. coli. The results are here. They're at the bottom there, but they put them down as observations. Kind of small for me to see, but hopefully you can see that they basically listed the different plates, and some say bacterial growth, and some say no bacterial growth. That's good, but it could be better, right? You can quantify bacterial growth. At least you can if there's a manageable number of colonies. You can actually count them. But even if you can't count them, if there were, say, too many colonies to count, what you could do is take pictures of them, and then at least that would be clear. So I would improve that. Suggestions for improving the results would be to actually give me images as opposed to just grew and not grew. Also, a table would be nice here, showing me what grew and what didn't grow, either as plus minus, or again, better would be numbers. And then the last thing I would suggest for improving this would be to give me the class data. And that could just be in summary form, like the other groups' colors and whether they saw growth or not. The very final small thing, they should have had a page number at the bottom here. This is supposedly page 3, right? But it doesn't say that. You can put it at the bottom. You can put it top, right, left. I don't care. Just make sure you have a page number. Now, here's their discussion. Discussion and conclusions often get intermingled, and that's fine. You can use either one. So let's just read through it. We expected bacterial growth on one of the 2 selection plates, kanamycin versus ampicillin. Since bacteria grew on the can plate with one vector, we know the plasmid was incorporated into the bacteria. They grew in resistance to the antibiotic kanamycin. Likewise, since the plasmid transformation for XA50 grew on ampicillin and not kanamycin, we know that bacterial growth on the AMP plate was due to picking up plasmid that had antibiotic resistance. So hopefully you're familiar, but if not, I'll just remind you that plasmids often have marker genes on them, And in bacteria, these marker genes are typically genes that confer resistance to antibiotics. So in this experiment, they were transforming with two different plasmids. Those plasmids have funny names that you see them here. But the important point is those plasmids had different resistance markers. One conferred resistance to the drug ampicillin, and the other conferred resistance to kanamycin. And we knew that going in. Right? And so what they're saying is basically, since we got opposing growth when we transform one plasmid versus the other, one grew on can, one grew on amp, that argues that everything worked. And I agree. It is an argument that everything's going the way it should. It would have been better to say exactly which plasmid had which resistance gene. In other words, they're saying that the PGB-KT7 has can resistance because it's growing on can plates. But you could go to the internet and find the plasmid map for that plasmid, and sure enough, you'd see that it has the can resistant marker on it. So in other words, you could verify independently what resistance genes are on there using outside resources. So again, always try to use all the resources that are available to you. You don't have to just learn from things we do in lab. You could be using the computer and the internet to learn from things outside the lab and make sure they jive. The next thing I would say is be careful with the word no. That comes here. We know that the plasmid was incorporated into the bacteria. Well, you think you know. And I can think of another scenario, right? What if that antibiotic simply wasn't active? So you made the plate and poured the kanamycin in, but the kanamycin had been stored inappropriately, so it wasn't kanamycin anymore. So if the antibiotic's not working, you don't know anything. So again, you could put that in your discussion. It is formally possible that kanamycin was not active in the plates. But then again, you want to rank these possibilities. I'd say that's pretty unlikely. And the reason it's unlikely is the other transformation showed the opposite result, right? It was sensitive to kanamycin. So in other words, you basically have an internal control in this experiment. Because you have two bacteria behaving differently on the two plates, that would argue that the plates really are different and the drugs are working. You could bolster that argument even more if you had the class data, because I happen to remember this, and the class data all fell in exactly this way. They all had growth and non-growth. And so, again, that's why the class data can be really useful. And what if your data was inverted to all the class data? Right? You could either say, well, all the class is wrong, or the simplest hypothesis, you somehow mixed up your tubes, everybody else did it right. So again, that's why class data can be useful. I'm not saying that will happen, but it's at least an argument that you can use. Then remember, one part of your discussion section should also be, what would I do next? What would I do in the future? Well, future controls. What if we put bacteria on these plates that had known resistance? So rather than doing the transformation and relying that as a control, we could have started with known resistant bacteria as a way to verify that the antibiotics were working. We could have also prepared plates that had both antibiotics. You can think about what you would expect to have happen if you had both can and amp in the same plate. Think about it. Should you get growth? Not growth? What do you think? The answer is you should get no growth, right? Because now you're providing both drugs, and the plasmid only provides resistance to one drug. So nothing would grow. You could also do a plate with no antibiotics. Think about what would happen there. Should you get growth or no growth? The answer is you should get tons of growth without antibiotic, because now you're not demanding the plasmid at all. But that's actually a really useful control. We often do that one. Because what if the observation had been that nothing grew anywhere? If that had been what that was observed, then you're left with all sorts of possibilities, one of which is that the cells are actually dead. Okay? So by doing a no antibiotic control, you could at least eliminate the dead cell explanation. What might you do in the future? Well, in this case, the future experiment was what we actually did, but you could just propose it in the lab. You could go on now, if you really want to make sure the plasmids are in these bacteria, you should be able to grow these bacteria up further, re-isolate the plasmids, and then test them in various ways to show that they are what you think they are. So in other words, this lab was not an end point. Putting the plasmids into the bacteria was actually a starting point for future labs where we're going to get the DNA back out and perhaps manipulate it. And that's a point I should stress. That'll happen a lot in our classes where we're doing labs and procedures to sort of get us to a final state. So not every lab, sadly, is answering a question, like an important biological question. Sometimes it's like cooking. You're preparing a component for your meal, but it's not the whole meal.